This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 129, recorded on April 12th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast where we explore virology. Joining me today from Western Massachusetts is Alan Dove.
Good to be here.
How are you, Alan?
Doing okay. How about you?
I'm just fine. It's a little cloudy, and the temperature, I don't know what it is in centigrade, to be honest. So
but who cares?
I'm not even going to say it, the temperature, because as we'll see later, we've been asked to report it in centigrade. But it's cloudy. And joining me today, right here in the TWiV studio in New York City for the first time ever, is Rich Condit.
Howdy, Vincent. Hi,
Rich,
everybody.
How you doing, Rich?
I'm doing really good.
Now,
this is great.
What are you doing here in my office?
I drove all of the way up here specifically to do a TWiV. That's what.
He did. Isn't he dedicated, folks?
You bet. We even added the TWiV studio as a place on Facebook. Okay.
Now, what does that do?
Well, it means that if you're ever in the neighborhood, you just pull out your iPhone, boot Facebook, okay, and you can hit check in, and it should pop up TWiV Studio, one of the places you can check in.
Right? And it'll show up on your Facebook page.
It'll show up on your Facebook page.
Cool.
This is important.
Nice.
You know?
I'll have to get an iPhone. I really do. That sounds great. Well, it's good to have you here.
It's great to be
studio.
Great to be here.
It's really
where
cool.
It's, what, 295 Kelvin or something like that, I think?
That's right. Yeah, that's right.
Well, it's somewhere in the 500s Rankine here.
Uh-huh.
No, we could do the conversion on Google, but we're not going to do that now. It's cloudy today here. Let's just say that. But spring appears to have arrived. And today we're recording on a special day because Rich is visiting New York, and we decided to do an all-email episode.
So that I don't have to cram.
Yeah, so Rich doesn't have to cram. Sometimes you should just go in and wing it.
I was thinking of doing that today, actually, but then I cheated and I went and I read the emails, and I'm just as glad because there's some stuff here that I really needed to see before.
But you know, Rich, what was I going to say?
Something about spontaneity?
Coming right after, we're just a few days, we just did TWiV on Friday. It's actually good to do email rather than 3 more stories.
Yeah.
In a way, so.
Yeah. Well, it's time. We got a lot of email,
we have a lot,
too.
Yeah, and there are more. So let's start. We'll alternate. I'm going to let you do the first one, Rich, because this is yours.
Okay.
Yeah, you can't be doing that, dude.
I know. I broke it. See? No, I was just putting it back together. Okay.
Rich has got this set of high-powered magnets, which you can use to make a viral capsid, a nucleocapsid, a helical nucleocapsid, and he's winding and unwinding them.
We could actually take a picture of this and use it for the picture.
Do you have your iPhone with you?
Yeah.
Yeah, I think later, let's remember.
I have my iPhone with me?
I'll take a picture of you doing that. We'll put it on the fan page. All right, go ahead.
Bryce writes, I enjoyed your discussion of the molecular therapy paper in this week's TWiV. That's TWiV 116, the paper where the authors put a cocaine analog onto adenovirus and showed that they could immunize mice against cocaine with it, right?
Yeah.
My lab and others have worked on using virus particles as scaffolds to increase the immunogenicity of various targets for quite some time. One thing that I wanted to point out and to correct one of the comments of Rich, I believe, is that this strategy actually is effective at targeting self-antigens. We can efficiently overcome the mechanisms of B-cell tolerance by displaying self-antigens on virus particles. My lab has done a number of studies in animal models, and a Swiss company, Cytos, CYTOS, has actually run clinical trials of virus particle-based vaccines targeting self-molecules involved in various diseases, Alzheimer's and hypertension. Thanks again for the enjoyable podcast. So, yeah, that would be, I'd like to see, well, of course it's not necessary, well, I guess, it would be a TWiV, right? Because they're using virus particles.
Sure.
Yeah.
So that would be a very handy application because use it to treat autoimmune diseases, I suppose.
Or to cause them.
Or to cause, yeah.
It's something you'd have to obviously manage fairly carefully because B-cell tolerance is there for a reason.
Right.
But for something like Alzheimer's in particular, it may make perfect sense.
Bryce is a professor at the University of New Mexico.
Right.
We'll put a link to his lab page. I wonder if he knows Michelle.
I was wondering the same thing.
Osmond, because she's also there, right? Unless there's more than one University of New Mexico. Let me click on his link, and we'll find out. Oh, the page cannot be found. In the time since Bryce sent this email, his page has gone down. Oop, it's gone. All right. Anyway, Bryce, do you know Michelle? If not, you got two TWiVists there at UNM. All right, the next one is from Jim, our friend in Virginia, who sends us a link to an article. Brandeis study shows economic impact of dengue virus in the Americas. And this is an article published in the American Journal of Tropical Medicine that costs the economic burden of dengue in the Western Hemisphere, $2.1 billion per year. This surpasses the loss from other viral illnesses on a country-by-country basis, including HPV, the most prevalent sexually transmitted infection, and rotavirus.
Wow.
So it's a nice article which summarizes this study. So we will pass on the link to our listeners. And thanks, Jim, for that. Yeah, these are expensive.
Yeah, and they say for dengue, it's mostly the indirect costs. It's not so much the treatment, which of course is just supportive.
Yeah.
It's the lost work and all that sort of thing.
Right. Okay, Alan, go for the next one.
So Daniel writes, I'm a graduate student in biophysics who discovered your podcast in December after finding your blog, all while working on a term paper about Ebola virus vaccines for my mammalian viruses biochemistry course. Despite my physics background, I've been interested in viruses since reading The Hot Zone in junior high. Oh, wow, I feel old.
When did you read it, in college?
Yeah, graduate school, actually.
Hey, you should have been working.
Yeah, I probably should have been. He continues, I just listened to TWiV 119, and after looking at some of the comments posted below 119, I've wondered what consideration has been taken to produce a vaccine based on a killed virus via formalin or oxidizing radiation or attenuation. But I must be jumping ahead since I've not done any journal search about whether or not the virus has been cultured. I assume he's talking about XMRV.
Yeah,
right?
that's my assumption.
The comments after episode 119, I had to go check this. They centered principally on that. Well, I'll just finish the letter and then we can address it. I've been working backward through the TWiV archive and have yet to get to the double digits. But so far, I do not recall if you and the other hosts have mentioned the topic about XMRV. If XMRV has crossed the species barrier into humans, then it seems likely that most of the population would have antibodies against it, much like the Epstein-Barr virus, given that mice follow humans everywhere. There are no rats in Alberta, apparently. Good show overall. Keep up the good work. A.
Is it true there are no rats in Alberta? How can that
I don't
be?
know.
I doubt it.
Got to be rats in Alberta.
Rats everywhere, right?
Yeah. And anywhere there are people, there are rats, right?
Yeah. Well, I mean,
maybe it's like snakes in Ireland or something.
We have talked about XMRV a lot, although not vaccines, because that's not even
several steps down the line.
Right.
Yeah.
Several orders of magnitude, probably.
Yeah.
Right. So as we pointed out, I think, last time I was on 2 weeks ago, it hasn't even been proven that XMRV is a virus that infects humans.
Yeah.
Right.
We're still at that point. So
we got a ways to go.
Talking about vaccines, not quite yet.
Yeah, you have to show that it infects humans, and then you have to show that it causes something when it infects humans.
Something he says here I want to correct. He says, if XMRV has crossed the species barrier into humans, then it seems likely that most of the population would have antibodies against it. You wouldn't make that conclusion.
No.
Not at all. I mean, Ebola has crossed over, but most people don't have
As a matter of fact,
antibodies.
Quite often, I don't know if you can generalize to say usually, but quite often, in the first instance of a virus crossing a species barrier, it's not transmissible from organism to animal in the new species.
Right.
Okay.
Right. Hantavirus would be another
Hantavirus,
example.
great example.
Mouse virus. Yeah, mice and humans are everywhere together, but not so much transmission of that.
Nipah virus is not effectively
Nipah.
transmitted from
Right.
person to person. You get it from your palm date sap. Remember
You
that?
get it from your date.
Yes.
Oh, yeah, right. Not yet. All right, next one's from Peter Rich.
Peter writes, congratulations on your great netcast. I will shortly commence my virology course for 2011, and I have 165 students, up from 74 last year. I'm thrilled and delighted to have discovered TWiV, which covers so much of the exciting, stimulating stuff that I talk about to my students. I will be telling them to go straight to your site, so please keep it up. By the way, if either of you are ever in Adelaide, Australia, let me know. You will be in grave danger of being dragged in to talk to my students to tell them just how fascinating viruses are. With best regards, Peter, who's a lecturer at the School of Biological Sciences at Flinders University, Bedford Park, South Australia. Well, I'm packing my bags.
Yeah, you want
I'm ready to go.
Sure.
Another road trip.
That's great. He has so many more students.
Yeah.
Over doubled. I found the same thing. I have 70 students this year, up from about 50 last year.
Wow.
So it's virology.
Must be doing something right.
I think it's TWiV. Yeah, it's infectious.
Think so?
Infectious enthusiasm.
Well, good.
The next am I next? I can't
I
even
think it's your
remember.
turn.
I can't remember this. John, right. So John is an MD in Nebraska. With regard to Nebraska, my individual 30-year radiology experience, many chest x-rays, is cavitary tuberculosis, 5 cases, lung cancer, 300 cases. Your personal experience based on knowing many more lung cancer victims than TB victims is verified. This is great. This is a great MD message. So on
Yes.
TWiV 119, which is me and Dave Tuller, the journalist, I think at some point we're talking about how the prevalence of infectious diseases and diseases in general, and we're talking, and I think we mentioned that probably in Nebraska you don't see much tuberculosis. And so he verifies that, more lung cancer than tuberculosis. So that's what that's about. All right, Alan.
Vince writes, presumably not you.
No.
Anyway, he writes, hello. This week in science, there was an article at the bottom concerning a phenomenal display of virus host evolution. And he actually links to a paper that we did on TWiV already because we're way behind on our letters.
That's right.
So his description of it is quite good. This study found metastasizing Leishmania parasites actually harbor a virus that stimulates the immune system, creating an inflated inflammatory response in the human host, which has been seen to lead to a higher reproductive success for the parasite. Wow. From a wider lens in virology, this may not seem so surprising as in bacteria, this is more common. Phage can transfer many beneficial genes to help bacteria to adapt to many diverse environments, including humans. Although in this particular case, we're talking about a different type of assistance, double-stranded RNA-mediated inflammatory enhancement. And then talks about an article on the Science News website, which I guess this was coverage of the same paper. Says the authors said this is the first reported case of a viral infection in a pathogen of this type leading to increased rather than reduced pathogenicity. This raises many questions concerning other protozoan parasites and whether their virus partners, exogenous or endogenous, assist their parasitic lifestyle. Certain virologists have long assumed that viruses assist symbiotic interactions between distant species. Louis Villarreal at UCI apparently believes this, where, for example, they extend gene addiction to a possible mechanism of mutualism generation. How widespread do you think this phenomenon is? Could viruses be used as defense, phage from our normal microbiome that possibly infect invading bacteria, or persistent viruses like TT viruses which possibly infect cellular pathogens? No evidence for that, however.
Well, I'd say it could be quite widespread.
Yes.
We just don't know. We have a lot of stuff to learn.
Yeah, absolutely.
Yeah, these are early days, but I wouldn't be surprised. Look at the virophage virus interaction to modulate algae growth. I don't know if you listened to the last TWiV yet, Rich.
On the way up from Washington to New York, yes, we listened to it in the car.
Yeah, we mentioned you a couple of times.
That's why I listened. I wanted to make sure.
that we mentioned. Anyway, that was a cool story, the virophage modulation of viral killing. And maybe there are many, many similar situations. We just don't know yet. And I think this is a great area of virology for the future, for now going forward, to look at the interactions between viruses and their hosts and what they do to pathogenicity. Sure.
I think we're going to have a better window on this with sequencing techniques, because we're now looking at a lot of viruses that don't necessarily cause disease, right? And turning up stuff that may have much more subtle effects.
Right, and now that we can amplify and sequence things without having to necessarily culture them, that also helps a lot.
Yeah, you can do surveys, right, and not have to culture because that biases whatever is there. Sure.
Right.
Yeah, it's a good point, Vince. Anyway, go check out TWiV 128. We talked all about that paper.
Vince is pretty clued in here. I wonder what he does for a living. He doesn't tell us.
What do you mean?
He's got,
Oh, I thought you were talking about me.
No, I'm not talking about you. You're Vincent. This guy is Vince.
Okay.
Right.
Yeah, he is clued in. It's good.
Yeah.
All right.
Next one is is that your turn?
That's my turn.
Good.
Benjamin writes, I noticed a set of strange coincidences related to your most recent podcast, TWiV 120, related to Egypt. Here they are. 1. Your podcast was recorded on February 11th, the day Mubarak stepped down. 2. You recorded the podcast in Alexandria, Virginia, which shares its name with the second largest city in Egypt. And 3. Your guest was named Ed Niles. I know, corny. Ben. I don't think it's a coincidence. I think that's all, written in the stars.
We could probably go ahead and tell Ben that we are, in fact, the Illuminati.
I think that was great. We got that the week after 120. I thought it was. I'm glad we're finally getting to it. It's really cool, Ben. Not corny at all. I love it. And I'm glad our listeners are thinking of this
tuned
stuff.
in, yeah.
Yeah.
It's important.
All right. Julie writes, thanks so much for the podcasts. I always learn a lot from you. I just finished listening to your interview with Dave Tuller, which I enjoyed. You made a number of confident statements about the certainty that science will figure out what's really going on with XMRV. I'd love to hear more about your perspective on Elaine de Freitas's work on an HTLV2-like retrovirus from a long time ago, because one of the discouraging things about what I've heard about that story is that it seemed like science didn't figure out what was really going on. I'd love to hear if you agree with that perspective or if there's more to the story than what I understand. Thanks so much for your work. Julie was a freelance math and science writer and a columnist for Science News and Wired. Julie, who did you hear that the story wasn't developed properly? Because, in fact, it really never went anywhere. So the paper was published in PNAS in 1991. Was entitled Retroviral Sequences Related to Human T Lymphotropic Virus Type 2 in Patients with Chronic Fatigue Immune Dysfunction Syndrome.
Senior author is somebody we've heard of, Hilary Kaprowski.
Hilary Kaprowski, Elaine de Freitas, the first author. So essentially what they did was to take patients with chronic fatigue syndrome and healthy patients, take their blood and look by a few assays for the presence of HTLV2. They did first an immunoblot. They ran out viral proteins on a gel, transferred the proteins to a blot, and then probed them with antibodies from the patients. Okay? And what they find in Western blots is that some viral proteins seem to be illuminated in the patient's serum, the serum from patients with chronic fatigue syndrome, but not from normal controls. Okay. Now, we don't know if these are specific for HTLV2. Some antibodies could cross-react. So that's my first criticism of that. And then she did PCR assays using virus-specific primers. And the PCR gels are very messy in this paper, but they do show products, DNA products in patients with chronic fatigue syndrome, but not in normal patients or healthy patients. But the products were not sequenced, so we really don't know what they are. So Dr. de Freitas never followed up on this observation. The CDC did publish a few papers looking into this, and they were unable to confirm the presence of HTLV2 in patients with chronic fatigue syndrome. And that was really the end of it. So I think science ran its course.
Right.
It was examined, and no one else found it. So I don't know why you would think that it would be discouraging.
Yeah, it's exactly kind of where we are with XMRV. It's an interesting preliminary finding as of 1991. And then subsequent work looked at it, wasn't able to confirm it, which, we're not necessarily there with XMRV yet, but that's the phase we're in right now, is subsequent groups looking at it and trying to figure out what's going on. And in this case, it turns out it's not reproducible.
Yeah.
My sense is that there may be a sort of a misunderstanding here in part of how this kind of stuff works, in that people may be thinking that you need to have proof that something is not true.
Right.
Okay? You have to prove the negative. And the fact is you can't prove a negative. All you can do is do your best to try and reproduce what some apparently positive result that somebody found. And if you can't reproduce it, all you can say is, I can't reproduce it. It's virtually impossible to prove the negative. You can say, I've looked everywhere I can, and I can't find it. Okay, so It apparently is not there.
Yep.
I don't know, maybe, Julie, you were looking for a story, but I'm glad you at least asked virologist. So
Yes.
You came to the right place instead of listening to the rumor mill. So there you go.
And actually, this whole HTLV story and the earlier EBV story is kind of instructive for folks who, look at the XMRV story and say, wow, this is unprecedented. Well, in fact, particularly in CFS, this is not unprecedented. This is the type of thing where people have repeatedly found viruses that looked like they might be associated with this disease. And, part of the skepticism in the virology community is that this isn't our first rodeo. We've been here before and it didn't pan out before. So the burden is on those who are advocating a particular hypothesis.
Well, and it's not just XMRV and CFS. This kind of stuff happens with lots of different diseases.
Yes, absolutely.
And lots of times there are, people say, I wonder if it could be this. Sometimes they get maybe a little bit of a sniff and then it doesn't follow up with verification. I think that there were a number of different agents suggested for cervical carcinoma.
Yep.
Before the data really started to look really good for human papillomavirus. And people still talk about other viruses perhaps being as cofactors in the cervical carcinoma. So this kind of stuff happens all the time.
Yeah, and AIDS.
Right.
Very early on, it wasn't at all clear what was causing it, and there were a whole bunch of theories, and there was preliminary evidence for several of them, and then the only thing that panned out was HIV. And so, you get a definitive answer, but all these false leads are lying around in the wreckage.
So I just want to leave one more thought with this letter. You say you made a number of confident statements about the certainty that science will figure out. So, Julie, that's based on working in science for 30 years. I've looked at it, and my conclusion is science figures it out. What do you think, Rich?
Yeah, I'll go with that. Okay, sometimes it takes a while.
Yes.
Yeah.
So I don't see why you would be skeptical about that, especially if you're not in science, so you haven't seen it directly. If you're looking from the outside, maybe you would get that view. What do you think, Alan?
Maybe. Although, this is not a new thing with science. Yeah. This is the way this process has occurred for generations and generations. The easiest example for most people to understand is nutrition. Because you're always seeing these headlines about this is good for you, this is bad for you, and then a week later, oh no, that other thing we said was good for you is actually bad for you, and the thing we said was bad for you. And nutrition is a field that's fraught with these kinds of difficulties because it's so difficult to do good experiments. And so, this goes back and forth, and you find a preliminary result, and it's worth following up on, and then a lot of them, in fact, the majority of them aren't going to pan out.
Yep. All right, let's move on to the next. That's for Alan, I believe, right?
Okay. Amit writes, Dear TWiV hosts, I enjoy your program for several weeks. I enjoy it very much, including TWIP. I find it very interesting, even for people who are not from the discipline. I wish to give something back, so I have several suggestions, which I think will make it even better. 1, the podcast is usually very long and unedited. It will be nicer if every episode will be specific for a limited number of topics which can be discussed during up to 1 hour. 2, the topic of discussion should be cleared at the beginning. New terms and methods should be explained at the first mention. Vincent does it in TWIP and maybe forgets it in TWiV because he's so familiar with the subject he forgets not everybody is. 3, no need for reading all emails in the podcast. Hopefully, I will start my own science podcast, the first Hebrew one, inspired by you and other podcasts on the sciencepodcasters.org site. And one last thing before wishing best regards and closing this letter, use Celsius degrees or Kelvin. Out of the U.S., no one can understand if it's cold or warm there. Best regards. Have a nice week. And Amit is at the Israel Institute of Technology in Haifa.
I don't know. We go on because we're excited about our topic. And we've done polls, and people don't seem to mind if it's an hour and a half.
And we like the spontaneity.
We like the spontaneity.
Yes. And we don't generally go over an hour and a half.
Yeah, we try not to. As far as explaining things, we probably could do a better idea. But that takes more time, Amit, and it would make it even longer sometimes. So
yeah, we do have to remember that. We got this.
Yeah, we do. That is a valid point. We should try to introduce things as we introduce them.
As for reading emails, I think that's the way we engage our listeners. And obviously they like it because we get so many. And so I would like to read them all just to show the wide range of listeners we have. And the questions are great.
Yeah, they really drive a lot of the discussion.
This show is based all on email. So you will see if and when you start your podcast, the best part is really getting questions and comments from listeners. So I could not imagine not reading all of them.
Yeah.
All right, the next one. Is that yours?
Is that me?
Sure, go ahead.
I think so. Yeah. Martin writes, and Martin is a frequent correspondent for us. Hi, Vincent. Here is a study showing transmission of yellow fever. The same yellow fever strain used in the vaccine is found to be infecting babies from mother's milk. Do you still think it is a good idea to vaccinate pregnant women? Best as always, Martin.
Oh, we have a guest in studio. Have a seat, Dickson.
I'm not a guest.
No, you're not a guest. You're a headphone.
You gonna mic him here?
Yeah. Clip on.
I am face to face with Dickson Despommier for the first time. This is really exciting.
Wow.
Listen, I'm excited.
But put the mic on before you. How was the Museum of Natural History?
It was crowded. We had a great time. This was their annual fundraiser dinner. So it was a panel discussion on all aspects of urban farming. And I got to meet the president of the museum, Ellen Futter. And then I sat at a table and I finally connected with an old colleague's son-in-law. Al Gold and I did a lot of research together back in the old days. And his son, you'll know him, was the one at the museum who bought Sue, the dinosaur.
Oh.
He bought the Tyrannosaurus Rex for the Museum of Natural History. So he came down and he knew I was there because obviously he saw me in the audience, and he came over to me at the table while we were having lunch and introduced himself. I had never met him before. So we had a wonderful conversation about dinosaur hunting. And
cool.
He's a mammalian guy now. He's after fossil mammals.
Excellent.
And viruses, of course.
All right, we just read an email about transmission of yellow fever. Interesting article here from the Canadian Medical Association Journal. Case report, probable transmission of vaccine strain of yellow fever virus to an infant via breast milk. So basically this woman was immunized with yellow fever vaccine. She was traveling and it was transmitted to her baby. Most likely the baby had some symptoms, recovered afterwards. I just want to point out that immunization of women with babies is not recommended with this particular vaccine.
Right.
And this is an example of why it should not be done.
Right.
In general, you don't want to conclude not to immunize women with all vaccines based on this. For example, women are very susceptible to influenza.
Yes.
And hence you should immunize them. So this simply underscores why we don't give yellow fever vaccine to breastfeeding women.
Right. And I think it's an important point is, on TWiV, we're big vaccine fans and we talk them up, but there are contraindications for some vaccines for some people. People who have egg allergies should not get an influenza vaccine that's grown in eggs. It's things like that. And pregnant women should not receive this vaccine for exactly the reason this paper outlines.
But was she pregnant when she got the vaccination?
No, she was breastfeeding when she got the vaccination.
Exactly. So my question would be, if she got it during her pregnancy, could the fetus become infected?
It doesn't say. All they do is say you don't immunize breastfeeding women because it can be the virus can be in the milk.
Right.
So fortunately, the baby was fine. But again, you shouldn't be doing this, and that just underscores it. So I don't think it's an indictment of all immunization of pregnant women at all.
Right.
Right. In fact, in the paper, they even point out that this idea that you shouldn't vaccinate pregnant or breastfeeding women it's a theoretical danger that the virus could be transmitted to the infant. And this paper proves that that was a valid contraindication, that
Right.
That in fact it can happen.
Yeah.
Who's next? I forgot. Is it me?
Gotta be.
David, our friend from the biology department at Vassar College, writes, I'm listening to TWiV number 118. One of the emails asked about virus-like particles used in vaccines, and you discussed that capsid proteins are capable of self-assembly. I believe it was Alan said it was like a bag of magnets coming together to form the structure. Here's a link to a video of magnetic capsid proteins, which, when shaken in a container, eventually form an icosahedral shell. I use it in my virology class to demonstrate what I mean by self-assembly.
Good heavens.
This is amazing.
Video is awesome.
This
Nice.
is amazing. I'm going to use it in my class too. I'm showing it to Dickson here right now.
And where, David, where did you get these? They're very cool. I'm wondering if they're custom made or something.
Yeah, tell us if you know, David.
There's a narrative that goes along with this that points out that it actually goes together as sub-assemblies.
Yeah.
Occasionally. Isn't that amazing?
Dixon likes it.
I love it. I don't like it. I love it.
Cool.
All right, so that's a cool video. I'm going to use that
All
too.
right, thanks, David.
Yeah.
Like a bag of
Actually, I have
magnets.
some magnets, David. I
Oh,
have different magnets, David, that I use too, and Rich has been playing with them incessantly. And I have a video I've made. I will post that one of these
Looks
days.
just like tobacco mosaic.
Yes, exactly. All right. Who is next, Rich?
Probably Dickson.
Yeah, Dickson,
I
you
don't
want to
think-
read an email
I don't
here?
think-
I do know how to read, and from a distance I can certainly read. Mike writes, I have an MS in biology and chemistry, but it is from the late 1960s. And I've been a self-employed programmer for the last 30 plus years after 10 years in a hospital lab. Many things have changed in the last 40 years, I imagine. I don't know if kudos is still used in protozoology, but it was my favorite text.
If it is still used, then congratulations are in order.
Stop it. Stop it.
Yeah.
I thought you'd probably say something along those lines. I began listening to TWiV to learn the current state of understanding of the XMRV because my late wife,
not
my
late.
wife, you better take it from here. I didn't bring my glasses in the room with me. Take it.
My wife has had fibromyalgia chronic fatigue for about 2.5 years. This was diagnosed at the Mayo Clinic in February 2010. I found your podcast dealing with XMRV interesting and educational, and have become hooked on TWiV and TWIP. Have my TWiV coffee mug ordered, and I'm in the process of listening to all the back episodes. I also wanted to let you know about the website bookfinder.com. In episode number 9, you mentioned a book named Fever by John G. Fuller. It is not currently in print, but you can get used copies through Bookfinder. I
Nice.
collect first edition speculative fiction, science fiction, and use this site daily. If it has been published, you can probably find it here.
Cool.
Love TWiV. Keep up the great wool, Mike.
Keep up
I
the
think he
great
means
wool.
work.
That's right.
Work, right?
I was wondering about that. Okay.
It does say wool, though. I can see it from here. It says wool.
I thought there was something here special that I didn't understand. I think we're doing great wool. Book, I figured, we're woolly or something like that.
It could be.
At any rate, bookfinder.com. I looked at that site. It is could be very handy.
Ah,
It'll find anything for you.
Cool.
nice.
All right. Are you up next, Alan? I don't remember.
I think I'm up next.
There you go.
Ilya writes, hello, a few weeks ago I defended my PhD thesis, and this is the photo of the last slide with gratitudes. Last line says, to Professor V.R. Racaniello for inspiration.
Oh.
And a couple of times when I thought my project was going to fail, you and all the wonderful people on TWiV indeed inspired me to carry on. Very
Neat.
cool, and he's at the Russian Academy of Medical Sciences.
Ah, terrific.
Chumakov's Institute of Poliomyelitis and Viral Encephalitis, which I know very well.
Cool.
Never been there, but very famous place. Chumakov, the father of Russian virology.
So who's the mother?
I don't know.
Can't have a father without a mother.
Chumakov's son is a virologist here at the FDA in the U.S. Konstantin, I know him quite well.
Very nice.
The next one is from Mark, who is professor at the University of Missouri, and he had originally written this to Welkin. That was a great episode. Welkin was on a couple of episodes ago. I love it when there are guests on TWiV that get into the oral history of different viral subjects. I am debating whether I should assign your intro about trim to my virology class this week. I wonder if anyone would object to having a podcast as an assignment. I'm already having them update virology entries on Wikipedia for extra credit.
Oh, well, that's neat.
Excellent.
That is neat.
I assigned my class this week some podcast listening for extra credit. So it's fine. Why not?
Yeah.
Vincent, if you ever have Alan Ryan on again, you should ask him to tell you about how the word pseudotyping was coined. It's another really interesting science history story that I don't believe has been written down anywhere. All right. We'll ask him.
I don't know anything about this.
I don't either.
Okay, we'll have to ask him.
We'll ask him. Do you want to try another one, Dickson?
Yeah, if we can move the monitor a little bit closer.
Well, you're going to have to move your body a
the
little bit
aged
closer because we're out of
persons.
wires here.
Oh, we are?
Yeah.
All right. Penny writes, it's been reported on Facebook that neither Frank and Sandra Rischetti nor Judy Mikovits has been invited to the upcoming CRIO conference. The committee also rejected at least one of their abstracts on XMRV and immune dysfunction. While there will be discussion of XMRV at the conference, the emphasis will be on contamination and negative studies. The absence of Mikovits and the Rischettis means that they will not be able to offer even a critique of these arguments, let alone defend their own work. I have appreciated your programs on XMRV and the last one with David Tuller, even if sometimes I've been critical of things you've said. But how much faith can a layperson like me have in the scientific process, in quotes, if important views are marginalized, rather? Where does the debate take place, if not at a meeting like this? Thanks.
This has been floating around for a while, and I really wish people would put a stop to it. There's this notion that certain people weren't invited to the CROI meeting, the big retrovirology meeting that just took place, I think about 2 months ago. Yeah, we're a little behind on our letters, so this was the upcoming CROI conference. Any retrovirologist can just fill out a form, cut a check, and go to the conference. It's like most scientific conferences. And this actually came up in the blog comments of a summary somebody was writing up of the conference. Someone jumped in and said, well, why wasn't Dr. Mikovits invited? And the upshot of this was the original blogger emailed John Coffin, who organized the CROI conference. And this quote from John Coffin's reply is, "No researcher who registered for CROI before the deadline was forbidden to attend. Of the more than 4,000 participants at CROI, less than 100 were invited to speak, and none of the speakers on XMRV was invited. All the XMRV talks were selected from submitted abstracts." He goes on, "Submitted abstracts are confidential, as is their review, so I cannot discuss any specifics of the process, except to note that only about half of them were accepted for presentation this year." So, in other words, if Drs. Rischetti and Mikovits didn't receive a gold leaf invitation, that does not mean that they were excluded in any way. That just simply means that they were in the majority of attendees, would have been in the majority of attendees who were not invited.
Yeah.
And they were free to submit an abstract. If it's true that their abstract was rejected, then they're in probably still the majority with half the other people who were there. So they could have gone, they chose not to, and as far as why they chose not to, you'd have to ask them.
Was there a poster session that they could have participated in?
Yeah, there were poster sessions. I don't know if the abstract, if nobody knows what actually transpired here. If they submitted an abstract, if it was rejected as a talk but accepted as a poster, nobody knows the story. But the truth is, had they wanted to attend, they could have.
Got it.
I think it's also worth pointing out that, at least in this discipline, it's not true in all disciplines, by and large, the scientific conferences are not peer-reviewed as journals. Okay, there is usually a panel that looks over the abstracts and decides, which abstracts might be most interesting for a presentation. But where the real peer review, the hardcore evaluation of science goes on, that's in the journals. Okay,
Yeah,
right.
And so that's, although the conferences are good for an exchange of ideas, in the end, the real documentation, the real peer review goes on in the written literature.
Something like this would be a perfect subject for a Gordon conference. Perhaps a morning session at a Gordon conference where nothing is published and everything is allowed to be said. Because I've run some of those Gordon conferences, and we've had some controversial subjects like the cause of multiple drug-resistant malaria, for instance. What is the actual genetics of that? We had two completely different points of view with good evidence in either direction. They kept bickering and fighting with each other, so eventually we just invited them to the meeting, and we gave them the entire morning session to fight it out with data. And that's how you do these things, and this would be a perfect ploy for that one.
It's a good idea, Dickson.
Maybe.
I think get them all together in one room. Sure.
Exactly right, and let them just yell and scream at each other and then accuse each other of not doing the right experiments, and then perhaps, as a result, you will do the right experiments.
Unfortunately, with meetings, many people feel they should be invited, so they don't go otherwise.
No, I understand that.
Right, which I think is unfortunate, right?
It is.
If you have something to say, you should apply to a meeting and go to it.
A little bit of self-pride there is too much.
Yeah, and at least if you choose not to go, don't go around saying that you were banned.
They weren't banned, that's for sure.
No.
Yeah, that's right.
Okay, that's good. That blog post is interesting there with Coffin's comment. Good. Rich, I think you're next.
Michael writes, Dear TWiV team, my name is Michael, and I'm on the faculty at SUNY Downstate in the School of Public Health in the Department of Epidemiology and Biostatistics. I've enjoyed listening to your podcast for quite some time. I find TWiV, TWIP, and now TWIM very engaging. I really appreciate the personalities of each of you four, Vincent, Dickson, Rich, and Alan, and the casual yet unflinching approach taken with each topic. I'm hearing trumpets. This is great. As an epidemiologist, one of my primary areas of research is the interface between infectious disease, climate, and geography.
Oh, good.
I use the application of GIS methods frequently in my work, and that is GIS stands for
Geographical Information Systems.
Okay.
It's like a hybrid between a map and a database.
Okay.
Right.
I use the application of GIS methods frequently in my work and was quite interested by a recent paper in the Journal of Tropical Medicine and Hygiene reporting on land use and West Nile virus. This is for you, Dickson.
Yeah.
A focus of some of my own work here in New York State. I found the results interesting, though as a skeptic was a bias-wary epidemiologist, I think it is difficult to draw inference from county-level data of West Nile virus cases.
Here.
And as we all know, correlation does not equal causation. Here again. Mm-hmm. When I was reading this paper, I immediately thought of Dickson. I have listened to many of the discussions you've had on TWiV regarding West Nile virus, and I've always quite appreciated Dickson's point of view with respect to the complex ecology of this infection. For example, I think the hypothesis that increased incidence of human West Nile virus infection can result from drier weather that provokes greater bird dispersal, leaving humans as a more available mosquito host, is well-founded. I would like to see or undertake some better studies that combine population-based epidemiology and multi-species ecology to get a better understanding of this process. As regards the paper, I'd like to hear Dickson's opinion on the juxtaposition of an eastern and western West Nile virus in the United States, based on the former being an urban disease and the latter being a rural disease, as described by the authors. The authors posit that regional differences in the distribution of human West Nile virus incidence between the eastern U.S. and the western U.S. can be delineated by distinctions in land cover across these two regions, developed/urban versus rural land. Moreover, they suggest that these land cover differences correspond to similar differences in the distributions of Culex pipiens and C. tarsalis between the two regions. The authors attribute differences in West Nile virus incidence to the exploitation of different local environments by the two Culecine mosquitoes. I am interested to hear your thoughts, particularly with respect to how such regional distinctions, if true, may fit with the overall dissemination of the virus from east to west across the United States from 1999 to 2006. I am wary of the aggregate data which were used to measure incidence, and we also do not get a clear picture of the finer resolution of the mosquito and bird ecologies that must be at play. Nevertheless, I do think this is an interesting hypothesis paper. Keep up the excellent quality of your podcasts. All four of you provide a true gem to anyone interested in virology, infectious disease, or science in general. You do a great public service, and I, for one, very much appreciate your efforts. Trumpets again. This is great. Also, I have a blog on infectious diseases called Infection Landscapes, and he gives the website. The idea behind this blog is to discuss the epidemiology and ecology and physical and social landscapes of infectious disease. Have a look and see what you think. I do weekly postings. Cheers, Mike.
Maybe we should have him on our show.
Yeah, that would be great. Want to come on, Mike, talk about it?
Yeah, I'll have to go back and read this paper. I mean, I'm not familiar with the details of the paper, but I accept your criticism because I attended a meeting in Washington, D.C. once, sponsored by the Institute of Medicine, in which they were looking at invading species. And, of course, West Nile virus is an invading species of virus, at least. But the mosquito also is an invading species, but it happened many, many, many hundreds of years ago, probably. At this meeting, it was clear that you could not obtain moment-to-moment epidemiologic data as to the outbreaks, and you could not determine with a GIS exactly where that case occurred. That data is being sequestered by the CDC and by the state public health officials, and they're not releasing it to anybody. And I don't know the exact reasons. One can only speculate, so I'm not going to do that. But I just say that I totally agree that if you don't have access to the exact pinpointed locations, you can't talk about ecotons. And in that sense, you can't talk about whether there's a distribution difference between eastern and western anythings. This is a peridomestic series of breeding mosquitoes. I mean, the Torsalis and Culex pipiens both breed near people and both determine their livelihood from polluted waters. So it's going to be difficult to sort these things out, but it's an interesting hypothesis, but I'd have to go back and read the papers in detail to make sure of what I'm saying before I start saying something.
Okay.
Sorry, I just erased that link there. I'm a PC guy in a Mac world here, and I'm having my difficulties.
You erased the link?
Yeah, that link he sent. I tried to copy and paste it.
I'll just do Apple Z.
Apple Z?
So Apple and Z.
It's back. Okay, great.
Right.
Oh, okay. By the time we get to the end of this, I'll have this Mac. I used to be a Mac freak. Okay, but UF beat it out of me.
Yeah, that can happen. Am I next? I believe so. I'll take it.
I'll take the one after that.
Okay. The next one is from Garen. Greeting. He wants some more trumpets. Greeting, semi-divine TWiV hosts.
Well, only semi.
Hey, I resent that semi.
That's okay.
What do you mean semi?
We got to get to like episode 200 or something like that.
Right.
Yeah, Dickson, just semi, not divine. I have a quick podcast recommendation for everybody. We all want more science, so what I would like to recommend is basically what TWiE should be this week in engineering. The podcast is called Omega Tau and can be found in a link. The host is German, but is kind enough to do a fair number of really interesting English podcasts. The show is a bit like Futures in Biotech in style, i.e. interviews versus panel. Topics range from quantum computing to flying the space shuttle to electron microscopes. Check it out.
Neat.
Cool.
So I did check it out. I really like it. And I emailed the author, Matthias, I think is his name. And I said, hey, we do these cool virus podcasts.
Yeah.
And he said, hey, you want to come on my show sometime?
Right.
He said, bring your host with you. I said, sure, let's pick a date. And he wants to do a show on bacteria and viruses. So I said, yeah, we'll do it.
What happened to the protozoans and the worms?
Wow, actually, these topics look great. Flying the space shuttle, and right below that, electron microscopes.
Yeah, he's a pretty wide-ranging science.
Actually, I learned a lot by that movie Space Cowboys about learning how to fly the space shuttle. That was a pretty good movie, I thought.
Deep wreck diving.
Oh, yeah.
That's one of my hobbies.
Oh, yeah. This looks great.
Yeah.
Covers the waterfront, so to speak.
Deep wreck diving, Alan? For real?
Well, depends on your definition of deep. I don't do the serious technical, and I don't do penetration dives. But these guys, John Chatterton is legendary. He's and crazy, in my opinion.
Yeah, a lot of those deep diving guys are crazy.
Yeah, that guy John Chatterton is one of the guys who discovered and identified the U-869 off the Jersey coast.
Okay.
Goodness.
Yes, I've read that book.
Yeah, it's
That's
in
quite
like
something.
220 feet of water, and it just.
That's about the limit, isn't it?
I don't do that. No, that's way beyond the limit.
Okay.
Yeah.
I would accept that. Have you been to Truck, Alan?
No, I have not. That'd be great, though.
I'm told it is.
Dixon, you want to take the next one?
Sure.
Where are we?
Manuel.
Manuel.
Okay, Manuel writes, Dear TWiV, could it be that viruses are implied in a wider range of diseases than we currently think? Science Daily published an article today where they stated that ALS could be caused by a human retrovirus. I know that we are in a discussion right now where XMRV could turn out to be not the case of CFS or some other diseases, but this does not have to be true for other diseases and retroviruses. Please keep up the great work, and I'd love to hear you guys talk about it. Best wishes, Manuel.
All right, I took a look at this paper. Now, Manuel, they don't say that it could be caused by a human retrovirus. What they make is the very interesting observation that in patients with ALS, amyotrophic lateral sclerosis,
Or Lou Gehrig's disease.
Lou Gehrig's disease,
That's right.
these patients seem to have more expression of an endogenous human retrovirus called HERV-K. So they got this information by looking at brain tissue obtained at autopsy, unfortunately. They compared the expression of one of the viral proteins, the Pol gene, the reverse transcriptase, with the same tissues from people without the disease. They have accidental deaths, so you can get brain autopsies from them, and also other diseases where the people die. So remember, we all have endogenous retroviruses in us. Most of them don't make, none of them make infectious viruses. Some of them make proteins and some of them make particles. HERV-K is one that from now and then does make particles, but they're not infectious. And what they find in this paper is that these patients with ALS make more RNA and more protein of the reverse transcriptase gene of this virus than other people. So it's intriguing.
Yeah.
We don't know if it's causative or a cause of, a result of the disease.
Is it tissue specific?
Well, they only looked in the brain
Right.
in this paper. So I don't know the answer. That is a good question, right?
Good question.
They looked in prefrontal cortex, motor cortex, sensory cortex, and occipital cortex.
And is it even, I didn't see the chart there, is it evenly distributed throughout those tissues?
No, it's differentially expressed,
Okay.
It's more in some places than others.
And which area of the brain is more affected by ALS?
I believe I don't know. I would be guessing.
Okay.
I thought it was a motor cortex disease.
Yeah, I think of it as a motor disease, but I don't know either.
But if you look at this chart here, Dickson, you see these dots are the
which chart?
figure 1A.
Figure 1A.
You see the dots are ALS. There's a lot of HERV-K polymerase expression.
Got it.
And it's down in systemic disease and non-existent in accidental and Parkinson's. So this is very interesting, and I think it's a cool observation, but obviously we need to do a lot more work to sort out what's going on here.
Sure.
Now, he says the protein is also made, so it's not a complete protein. We don't know if it's actually encoding an active enzyme. They really need to do that next. That would be interesting, right?
Yeah.
And what
there's an ALS center here. At
there
Columbia.
is. There is, yeah.
In fact, our former dean, Alan Rosenfield, died from that
ALS, yeah.
very sad disease.
So gentlemen, what would we have to do next with this?
Well, they have patients at our clinic that are at various stages of their disease.
Mm-hmm.
So what you could recommend, and I know it would be invasive, unless it's in other tissues, is why I asked. But apparently, it's not even evenly distributed throughout the brain tissue. So it's in specific areas. It looks like. I know the director of that clinic, and I'm sure he would be very interested to hear this.
So they conclude these findings identify novel HERV-K and genomic markers of ALS, which may have important implications. The mechanisms by which these viruses cause or contribute to changes requires further study.
Now remember, every patient here is a patient who expired.
Yeah, these are all deaths, right?
So there's no brain biopsies or anything else that this takes advantage of. But I wonder if, to make the original diagnosis of ALS, they had to take a biopsy.
I don't know the answer to
And
that.
if that's true, then they could go back and look at
Sure.
the tissue bank and see whether it was there to begin with.
Yeah, unless you had some kind of in vivo method for detecting this thing.
It would be interesting. I find this fascinating observation. It
Yeah.
requires a lot of follow-up, but wouldn't it be interesting if reactivation of an endogenous retrovirus in some way was related to this disease?
Yeah.
Sure.
It would be.
Sure.
Is there a good animal model for ALS, or even
No.
any animal model?
Don't
No,
think so.
no, there is not. That's the
Because that would
problem.
simplify things a
Yeah.
bit. But just a note on Science Daily, the site, they essentially reprint press releases.
Mhm.
Got it.
So when people are reading it, they shouldn't draw major conclusions from what's presented on Science Daily. They should actually go to the original paper.
And then write us a letter.
Yes.
Right, exactly.
I went right to the. I usually, when people send links, I always go to the original article.
Right.
Yeah.
I don't want to read the press release
Yep.
because, for what Alan said, but also it gets me a little upset when I read press releases.
They are often not so great.
Yeah.
This is also, I'm not dinging the study here, but it's a fairly small
Yeah.
number. 28 ALS patients, 12 with chronic systemic illness, etc. And there's others here. So one of the things that needs to be done is a much larger study in other places.
Yeah.
Sure.
I would look in other diseases as well to see if
Well,
there's any.
there's a lot of related diseases to ALS here, too.
Mhm.
Nancy Wexler's
Huntington's.
Huntington's.
Well,
A lot of these
they look at Parkinson. There's no correlation
are-
here.
Right.
So, but I think it's worth looking at other diseases to see whether there is a correlation with increased expression of these endogenous viruses. Who knows? Right? Even chronic fatigue syndrome patients. It would be interesting to look at this.
Mhm.
Right?
Why not?
Okay, the next one. Who is up next? I don't recall. Alan?
I'll take it.
Alan.
It's from Carrie.
Carrie writes, "Hello, Vincent, Dick, Alan, and Rich. I'm currently a graduate student in the Barrick Lab at UNC Chapel Hill in the Department of Microbiology and Immunology. I love listening to your podcast, being turned onto it by Eric Donaldson, whom you've had on to talk about the bat virome. Eric's work on bats is great, but equally great is his work with noroviruses. I may be a little bit biased working on noroviruses and all, but I think it's high time you do an episode on this virus since it affects pretty much everyone at one point or another. I like to call it the puking pooping virus. In the United States alone, over 23 million people get norovirus each year. I could go on and on about my virus, but I would rather have you guys do that on an upcoming episode. Thanks again for doing TWiV. I love it."
So we should have Carrie on.
Stay tuned, Carrie. It's
That's
coming
right.
up.
You're gonna
We
be
actually
sorry.
have somebody scheduled.
When
is it? May 19th or something like that? I
Yeah,
forget.
sometime in May. Yep, we have a neuro schedule on the schedule, so there you go, Carrie.
Yep.
We heard
Perfect.
you.
You get a real short one. That's not fair.
How do I pronounce this? Dieter?
Didier.
Dieter.
Didier.
DDA.
Writes, "Here is a new English music group, The Vaccines." And there's a link here that I checked out. "Can't be ignored by TWiV, maybe a pick of the week." And it is indeed a rock music group, and there's a couple of excerpts from their band. Good hard rock. I think it's appropriate for viruses.
I like
vaccines.
his little
Yeah.
signatory thing there.
Yeah.
That's
You
great.
can read the next one, too.
This Didier, by the way, is from France. And I believe Didier is written before.
Mm-hmm.
Thanks for that, Didier. Oh, yeah, the next person is near you, Alan.
Oh, yes.
Marcia writes, "Several weeks ago I had occasion to speak with Judy Mikowitz. I mentioned your podcast show to her, and she said that she and Frank," I assume she meant Frank Rossetti, "would be willing to be guests on TWiV. I think it would be fascinating to have both of them on the same show and to hear your usual free-ranging chat between you regulars and the two of them. From what I understand from our conversation, WPI has finally been able to raise enough funds to buy their own sequencer, and they have a lot of ideas about what they'd like to investigate in regards to XMRV, polymorphic forms, etc. I'm including the contact information for the Whitmore Peterson Institute below, in case that would be useful to you. In my experience trying to speak with Dr. Mikowitz, the, quote, guardians at the gate unquote, are diligent in screening her calls. If you don't have any luck arranging something through the media contact below, you might succeed by speaking to Dr. Mikovits directly, since she did clearly say in our conversation that she'd be interested in talking with you. I eventually got through to her by doing some name dropping. Dr. Klimas suggested that etc. I'm not proud, but whatever works. Disclaimer. Although I listen to all the episodes of TWiV and find them fascinating, I have a particular interest in XMRV because I tested positive for XMRV via serology. This was through WPI and participation in one of their studies. I have been ill for many years with CFS, and I follow as much as I can of the science relating to CFS. I don't know whether XMRV will turn out to be the answer, but it certainly is interesting. The work with spinal fluid proteins associated with CFS and fibromyalgia is also very promising. I was raised with a scientific background, and I hope very much that CFS will become understood and effective treatments available someday. Again, thank you very much for your interesting show. Sincerely, Marcia. Well, Marcia seems to have at least an open mind as regards this, and I appreciate that.
I would certainly be interested in having Mikovits and Richetti on. We could talk to them about this. I've tried to arrange something, but nothing's happened so far.
Yeah.
So it would be interesting because they are the first people to publish that. I think we'd love to have them here and have a free-ranging conversation.
Sure.
Right? So
You bet.
we'll see what happens. Dickson, do you want to do one, or is
Sure.
it too difficult for you?
It's too difficult for me. Too many sentences.
I'll take it.
No, I'll do it.
Oh.
Sophie writes, in parens, with respect to the decline, effect, and the scientific method. I think this article is quite disturbing. Even if the author is right, I have serious problems. I have a serious problem with the sentences like, if replication is what separates the rigor of science from the squishiness of pseudoscience, where do we put all these rigorously validated findings that can no longer be proved? Which results should we believe? It seems to me that it is quite a problem at the moment for the general public to distinguish between real science and more alternative things like homeopathy. And I think this article might give, as Mr. Mark Christlip puts it, the alternative wackaloos even more ammunition, even though it is unwarranted. Also, I think it is a big problem that, as Alan points out, the beginning is sensational, because if somebody doesn't read the article through, they might draw the wrong conclusions. Besides, that I find it really interesting, and I would like you guys to talk more about it. At least in Denmark, it isn't a problem. Most science students know about it, and it isn't discussed in class, and I think it should, to avoid bachelor students ending up finding out about this in their first really important paper/assignment. In general, I think there should be more focus on the problems with science, so we are prepared to handle these things before we get out into the real world, in quotes. We just had a case of a cheating brain scientist who now has been convicted of using research money for clothes and stuff, trying to blame this one on one of her students, and faking mouse experiments results. It was quite clear that the system isn't really geared towards handling these cases. Both this and the MRI autism study might have been caught way earlier if scientists were more aware of potential problems. Please discuss, and sorry for the rant. Sincerely, Sophie.
Well, we like rants to start with. That's fine. A lot of people rant for us, and we appreciate that.
This is all true.
I don't know if you were present for that discussion, Dickson.
I was not, so I'm unable to address it.
We talked about an article that appeared in the New Yorker several months ago about difficulties of scientific replication, especially in fields like behavioral science.
Got it.
Right.
I thought we did a good discussion, Sophie, and you point out a lot of good things yourself. So,
by the way, since I wasn't there, I can add something to that now. This guy, Judah Volkmann, who became world famous for his work on angiogenesis at Harvard, was for many years ridiculed behind his back because no one could replicate his results, and yet he was a very honest and straightforward man. So what he ended up doing was inviting people to come into his laboratory and work with him to see if they could get the same results that Judah Volkmann got, at least working in his space. And lo and behold, they did. So, I mean, they had something in their own lab that wasn't quite the same as Judah Volkmann's lab. And Judah Volkmann openly and willingly invited them in because he knew that what he had found was correct. So Just because you don't get it to work doesn't mean that the other guy didn't get it to work.
That kind of stuff does happen. You move labs, and all of a sudden, stuff doesn't work anymore.
You betcha.
That's what they actually talk about in this paper, in this article.
Sure.
They talk about an experiment that was set up in 5 different locations. They used all of the same experimental conditions, but they got different results.
Sure.
And because every lab is different.
That's right.
You know, so.
In fact, the strains of mice will be different. Everything is different. And,
right. And the issue is you can't necessarily control for everything, and especially when you're looking at small effect sizes, which is why this comes up most often in things like behavioral science.
Exactly.
If you have to do a lot of statistics, then you can also get things with significance chasing and kind of structuring the experiment to get the result that you expect to get, and that can really skew things.
Sure.
Alan, are you next?
I think I am.
All right.
Which, by the way, is why I did my thesis in a field where everything was at least 3 orders of magnitude, so I didn't have to worry about this.
Right.
Okay, so Ina writes, you don't meet many people named Ina these days.
No.
Hi, Vincent, Dick, Alan et al. In TWiV 70, you talked about host manipulation by outside invaders, viruses, parasites, etc. An article giving many examples of this type of manipulation can be found in Scientific American, March 2003. The author of this article is Robert Sapolsky, and it's entitled Bugs in the Brain. I enjoy TWiV immensely and was introduced to TWiV by a friend whose daughter took a virology course at Columbia University. Listening to TWiV was part of her assignments. I myself am an alumna of Barnard and have worked extensively in various areas of cardiovascular physiology. Keep up the great programs of TWiV and TWIP.
Nice.
Yeah. I used to coach the debate team at Barnard.
Really?
Yeah, cool. Barnard and Columbia undergrad.
Well, I must say there is only one virology course offered at Columbia.
I wonder who assigned that.
And you know who teaches it, right?
And I taught it, so I must have had that daughter. And yes, I did assign listening to TWiV. Absolutely.
So that's where we get our audience.
Well, it's only one semester a year, so there are other people who come in. I think it's my turn. Rich will like this question. Michael writes, first, I would like to say thank you for a great podcast. I am a parasitologist who recently started a job in virology, and I found TWiV after watching your lecture series, my Columbia lectures. Thanks for that, too. I drive for 2 to 3 hours a day, depending on traffic, and TWiV and TWIP have been keeping me sane as well as brushing up my science. By the way, I am now up to date with TWIP. 1 month is too long to wait for the next one. So my question relates to a statement you made about growing influenza in cell culture. You said that in order to get a high titer of virus from a cell monolayer, you had to start with a low MOI. This is a general question, but I work with avian influenza, and I can't understand why this would be the case. This is why. There are a limited number of cells in the monolayer. Influenza causes lysis of the cell, and presumably there is a limit to the number of virus particles produced by each cell. Therefore, the MOI will only affect how fast the final virus titer is reached. The variability in final virus titer may therefore be due to when the flasks are harvested. If I have got this wrong, I would gladly be corrected. I have not finished listening to all TWiV episodes, thank goodness, as it really does help with the driving. So you may have answered this question. Thanks, and keep up the good work.
Yeah, we discussed this a while ago. His description of the problem is, on the surface, correct. You know, if you've got a certain number of cells and eventually they all get infected and they yield the same amount of virus, it shouldn't matter what multiplicity you start with. However, it does, and the reason is the generation of what we call defective interfering particles. And the problem is that some viruses, if you passage at a high multiplicity of infection, will very easily throw off variants that are by themselves replication defective, but will replicate at high multiplicity because there's always enough good virus around to support the replication of the bad virus. And so what happens when you do that high multiplicity of infection is that the bad virus actually interferes with the replication of the good virus. You get a mixture of these defectives and the normal stuff around, and the titer crashes. It goes down. It's interesting. This was I think I already said it was called the von Magnus effect. It was discovered in flu, right, by von Magnus, the original paper, where he did serial undiluted passage of flu and saw with passage the titers fluctuate from high to low, because what happens is the titer goes so far down ultimately that when you're infecting, you're effectively infecting at a low multiplicity of infection, so that cells are really only infected with one viable particle, and then you get a reasonable yield. The titer goes back up. Now you're doing a high multiplicity infection. It crashes again. Not all viruses do this. Flu does it. Polyomavirus does it, I know. A few others do it.
Yeah, I had this experience with flu in the lab. You use an undiluted stock, you get very low titer on one pass. And it is not only because of DI particles, but the virus kills the cell very quickly when you put too much virus on. Same thing happens with adenovirus. Adenostocks, they have a lot of fiber protein in them. If you don't dilute it, it will just lyse the cell before you can have sufficient replication. So it's a combination of those two things.
Do viruses ever kill cells before they actually finish replicating, if there are too many to start with?
Yeah, the case of adenovirus, the fiber will penetrate the cell membrane and kill it. So that's why you need to dilute it for many viruses.
Okay.
All right, the next one is from John, who writes, Dear TWiV-tastic 4.
Cool.
The TWiV-tastic,
That'd be a good T-shirt.
The TWiV-tastic 4.
Yeah, the TWiV-tastic 4.
Don't forget the cape.
No capes. No capes.
Okay.
You guys don't know what that's from.
No.
No, what's that from?
Yeah, what is it from?
We'll let a listener tell us.
Okay.
If you tell me what No Capes is from, I'll send you a TWiV mug.
I can Google it right now. Alan,
No, you're
want it?
you're not going to do it.
Come on, do it.
You got TWiV mugs already. I must disclose that I thoroughly enjoyed this most recent episode, number 124. That was Viruses That Make You Better. I think it had a nice mix of detailed science, some insight into how science in the modern age works, a nice nuanced discussion of what to do about smallpox, and plenty of puns. If people think in any way similar to me, they think of some nebulous smallpox vials kept in cold storage somewhere, like the DNA was in Jurassic Park,
Right.
where security is as weak as having just one Wayne Knight as the IT guy. Obviously, that's a bit of a caricature, but I think you get the idea. When the listener email about funding came up in my head, I was already rambling similar to what Alan said, so I was quite pleased to hear him articulate my reactionary thoughts better than I was rambling internally. Speaking of Alan, I knew this would be a good episode due to a critical measure in TWiV quality. That is the time to pun measure. When Alan is in top form, it's nice and low. And there was Dick right at the end for the walk-off victory with the vertical farming quip.
I forget what I said.
You should listen. Dickson, you should go back and listen. So, in summary, great guests, great topics, great to have everyone back, and to know that Dick has no escape from the podcasting. Someone send him a fishing video game so he won't have to travel. Hey, Dickson, you hear that?
I hear it.
You have no escape from podcasting.
I hear it. I know I love doing this. You know I do.
The time to pun measure. We should actually quantify that.
Time to pun.
Yeah, the time for the beginning of the episode, right?
Yeah.
Yeah.
The TTP.
So when it's small, that means Alan is in good form.
That's right.
That's it.
Right. And does it matter what caliber of pun we use?
Well, there's no.
Oh, no.
Are you kidding? There is no caliber of pun.
That's a good point.
I've forgotten who we are up to. Rich?
I'm feeling the urge. Sven Urban writes, Gentlemen, in the last episode. Oh, by the way, why am I able to address you like that? Why not ladies and gentlemen? Sorry, that was a glitch. Let's reboot. Gentlemen, in the latest episode, I think we're having our chains pulled there.
Yes.
Indeed.
Justifiably. I mean, we have had some ladies on, but it's not 50%. Gentlemen, in the latest episode, as of this writing, number 124, you were once again talking about the possibility pending call for destruction of all known smallpox virus stocks. You also mentioned that nowadays, as the genetic code is known for some lines at least, it would be possible to reconstruct the virus from scratch, although it would be with some difficulties. Not that I'm about to try it at home, my wife might object, and she's both a better chemist and a better cook than I am anyway, but how would you go about it? And what would the largest hurdles be? Would it be done in test tubes and reactors, or would you enlist the aid of some poor doomed cell to do it for you? And would you really get proper healthy virus alive and kicking as a result? A somewhat thorough step-by-step overview would be much appreciated. As an aside, I would once again like to suggest a listener pick of the week, this time the 3 Science of Discworld books by Terry Pratchett, Ian Stewart, and Jack Cohen. First, The Science of Discworld. Second, The Science of Discworld 2, The Globe. Third, The Science of Discworld 3, Darwin's Watch. In these books, all available in paperback, the authors explore science, particularly physics, cosmology, and evolution, as seen through the eyes and sharp but weird minds of those denizens and wizards of Discworld. These books are good, and anyone who is at least reasonably familiar with science and happens to like Discworld will love these hilarious but also very serious books. And best regards from a thawing Sweden, Sven Urban. Very nice. I'm not familiar with the Discworld books.
Me neither.
Me either.
Okay, well, we'll have to fix that.
I like the idea of a listener pick of the week, though.
Yeah, well, we got quite a few in here. Oh,
Yeah.
we could have that as a regular deal.
We could do a listener pick each time, so
that.
we could pick an email with a listener pick. Yeah,
That's a good idea.
Nice.
Okay, so let's make smallpox here. I actually put a link to a paper that I think is the closest published sort of methods paper that gives you a clue of how to do this. Of course, it is now possible to reverse engineer, I think, virtually any virus. I recently researched this for something that I was writing, and kind of went through all the families.
I know what you're writing,
and I think they're all represented. Okay, I think you can reverse engineer any virus. The methods differ depending on what virus you're talking about, and it's a little peculiar, or not peculiar, but it's a little different in the case of poxviruses, because since they are DNA-containing viruses that replicate in the cytoplasm, the DNA is not infectious. The virus has to carry in all of the enzymes ordinarily required for replication. And in fact, also, those enzymes are complex enough. You're talking about, for early transcription, to launch an infection, you're talking about 20 different enzymes or something. So you can't just transfect in a bunch of plasmids and hope that it happens. What you really need is a helper virus. So you infect the cells with a helper virus that is essentially non-homologous with the virus you're trying to build. Okay, so for example, if you were trying to build smallpox, you'd probably infect with something like foulpox or a Leporipox, that is a virus of rabbits. Probably foulpox would be a legitimate helper, and then transfect in a whole lot of DNAs that you can just make synthetically. You could theoretically construct the entire genome from sequence in a cloned molecule and transfect that in and reactivate it. But what is done in this paper is to transfect in cloned molecules that are fairly large and overlap, and the enzymes provided from the helper catalyze the recombination of those with each other in the cell to assemble the whole genome, and the enzymes to initiate the transcription and launch the infection. And since the helper is not very homologous to the virus that you're trying to build, you hope that you get limited or no recombination between the helper and the transfected fragments. I think that still needs to be rigorously proven to find out how unfrequent that is, but my guess is that it is infrequent enough so that you could build smallpox. Now, the only remaining problem is the very ends. These are hairpin termini. They have an unusual structure, but you can also build clones of those and should be able to I forget how they did that in this paper, but you should be able to transfect those in and make it. So basically, helper virus, that provides the enzymes but won't recombine with the things you're transfecting in. Transfect in a bunch of overlapping synthetic DNAs, and nature will take care of the rest.
And you would do this in a cell culture.
In cell culture.
Small scale, right?
Small scale.
And then if you wanted to use this for nefarious purposes, you would scale it up.
Yes.
In reactors.
And talk about nefarious.
Very, very carefully about because of the possibility of killing yourself doing it.
I have a really gruesome idea here. Okay, if you out of that culture anticipated that you would get a variety of things, some of which might be pathogenic and others not, you could select for the one that was pathogenic by infecting somebody with the lysate. Okay, and recovering virus from that.
And that person would be sacrificed, right?
That person would be well, they have a 70% chance of living. Okay?
But there are people who would do that.
There are people who would do that. And then, in fact, yeah, you could grow it up in bioreactors. But I was just having this discussion the other day with Nile. You know, for the last several years, we've been growing 6 to 12 liters of infected cells a week in our laboratory. Okay, if we had harvested all of that and made virus from it, okay, this is vaccinia virus, of course, but even a fairly, we are not a big laboratory, okay? A fairly small operation could grow a scary amount of something like smallpox.
There you go, Sven Urban.
But it's a whole lot easier to smuggle box cutters onto an airliner or fill up a Ryder truck with ammonium nitrate and fuel oil, so...
Gentlemen, we are at 1 hour and 23 minutes.
No kidding.
Indeed.
Time flies.
Shall we do one more, or shall we go to picks of the week? What do you think?
Next one's kind of longish.
It is.
All right, let me just do one short one then. This is from David. Love the show. Absolutely one of the best overall podcasts. Great dynamic of science, humor, humanism, and camaraderie.
There
you go.
All right.
On the last episode, you played some examples of DNA music that I would like to acquire. Would you please send me the link? Couldn't find it in the site and the name of the composer, so I did. David, there you go.
Cool.
He's from Rockwell Automation.
Wow.
Oh.
I don't know what that does.
One can imagine.
All right.
All right, there you go. And we have so many more left here. I thought we'd get through them all.
I thought we were going to get further, yeah.
Yeah.
I rudely interrupted your routine.
No, it was only 5 minutes. It's fine. All right. Well, yes, there are a lot of good ones here. We will just have to do it next time. All right, let's do some picks of the week. Wait a minute. What is this?
I wrote that in there.
Oh, Dickson is here. You thought he wasn't going to be
here? Well, I was looking at... I was going over this morning, and I saw that you didn't have Dickson down for a pick. And I thought, do you mean to tell me I came all the way to New York City and Dickson's not going to be here? Then he showed up. That's great.
He came here for
but
you.
He doesn't have a pick.
That's it.
He doesn't have a pick.
That's all right, Dickson.
No, you know what my pick is? I just got back from the American Museum of Natural History. I'd recommend going there.
There you go.
This is a fantastic... I just met the president, Elaine Futter, and she's a terrific woman, and she's a go-getter, and she's committed to discussing and presenting issues of ecology through this museum. It is not a dead museum. It is a living museum. This is not just about bones and stuff.
They got dinosaurs, Dickson?
They've got the biggest dinosaur in the world going up right now. The biggest sauropod that ever lived is going up as a demonstration right now.
Oh, is that what I saw in the Times this morning?
Yes.
All right.
Yeah, so it's really right on target.
I can't believe living in a place where the New York Times is your daily paper. Isn't that wonderful?
It's great. That's absolutely great. I think she mentioned also that this dinosaur ate something like 100,000 pounds of food a day.
Wow.
You know, they probably had viruses, Dickson.
The calories.
Absolutely.
She said 100,000 calories a day. That was their... Oh, of course they had viruses. Of course they had
All right,
viruses.
that's Dickson pick.
That's my pick.
American Museum of Natural History.
That's right.
Unfortunately, most of our listeners are not going to be able to go, but if you are here, check it out.
And
it is a great museum. I haven't been there in a number of years.
As a little sidebar, I had the pleasure of sitting next to the guy who sold the Tyrannosaurus Rex skeleton called Sue. He was the one who purchased it for the museum, and I had the pleasure of sitting next to him.
You certainly are an elbow rubber.
It was fun today. You don't get these many opportunities, so
I never get them, but that's fun.
That's not true.
I like doing TWiV. I'm happy to do this. Rich, what do you have for us?
Last time I was on 2 weeks ago, I picked the most recent issue of Emerging Infectious Diseases had just come into my mailbox, and I picked up something from it right away, and it was only later that I realized that there's actually a significant number of pox papers in that issue, and including an engraving on the, or a reproduction on the cover of a painting of Edward Jenner vaccinating a child. And so I wanted to highlight actually that whole concept. There is one of the staff at the Emerging Infectious Diseases is a woman named, I'm going to butcher this name, Polzeni, Polyxeni, Polyxeni Potter. Okay, I apologize if I've butchered that, because I have enormous respect for this person who, for each issue, picks an artwork for the cover.
Nice.
And writes a little feature in the back of the magazine that describes the artist and
wonderful.
the painting, and usually tries to, and does a little art history on it, and tries to tie it in with the most recent issue. And they're fascinating. They're great. They're just about a page long. I hope someday she just publishes them all as a book.
Nice.
They must be available somehow. But at any rate, she has an interest in art and art history, and it's a great contribution to this magazine. I read them all when they come through. And in this one in particular, that's a painting by Eugene Ernst Hillermacher from the mid to late 19th century. Actually, 1884 is the painting, Edward Jenner vaccinating a boy. She has in this not only stuff about the painter and the painting, but she's got in a very short coverage the Jenner story, okay, which I thought is really nice for people to read. So I wanted to plug her work in general,
very nice.
And that article in particular.
Oh, nice.
Yep.
That's a cool pick. I like that. I like that.
Yep.
That cover. It's wonderful.
Mm-hmm.
Alan, what do you have?
Well, we're recording this show on April 12th, which is 2 spaceflight anniversaries. It's the 50th anniversary of Yuri Gagarin's flight, and I just saw today the Google Doodle is based on that. Mm-hmm. And it is the 30th anniversary of the first flight of the space shuttle.
Wow.
So I was in kind of a spacey mood. And
how interesting.
I've had this link kicking around for a little while, and it's something that's kind of an odd little bit of NASA history, the moon trees. This was one of the Apollo 14 astronauts got handed some tree seeds to take into space and to take to the moon. And he did. It was a mixture of pine, sycamore, Douglas fir, a few others. And the idea, of course, then, virtually nothing was known about the effects of space flight on biological systems. And so he took these seeds to the moon and back, and then they got sent around and given out to various organizations and planted. And these trees are growing all over the place. Cool. And there are people who've made it their hobby to go and find these moon trees and when they were planted and what their condition is. And so there's a website dedicated to this, and if you want, you can go and look for them. And I'm just looking down. There's one in New Jersey, and I'm not seeing any in New York.
There's one in Florida.
There's one in Florida. Yeah, there's actually
there's one in Gainesville.
Yeah, there's one in Gainesville.
There's a sycamore
at the University of Florida.
Well, I have to go check it out.
Well, come on, you guys are right next door. I mean, to the Space Center.
It is true. We've even got an American flag that went to the moon. I think there was a UF graduate who was an astronaut.
Nice.
So, Alan, today was the first shuttle 30 years ago?
I believe that's correct, yes.
Well, I have to say that on that day, an article describing my infectious polio clone was also in the New York Times.
Nice.
And they said to me, "We're sorry it didn't make the front page. We had to bump it off because of the shuttle launch." So it would have actually been on the front page, but for the shuttle.
Interesting.
And now I will also... So it was 30 years ago that I did that. How about that? Cool.
So and you talk about poor editorial judgment, because, now they're retiring the shuttles, but You're still working.
And the DNA lives on forever.
That's right.
DNA lives on. That's
So
elastic.
what other anniversary is this, Ellen? Sure you
It was
know.
also there was a bit of an altercation that took place 150 years ago.
Oh, that.
Yes, it was referred to by some of my relatives shortly afterwards as the recent unpleasantness. In some of their
Yeah, very good, Alan.
They're reenacting Fort Sumter sometime later this month as a way of commemorating that.
And not just in the House of Representatives.
And not just in that. That's correct.
Sorry.
With a 2-on-2 basketball game in the. This is exactly right.
Well, my pick of the week is actually Michael's website, which we read the article of. It's called Infection Landscapes. A wonderful blog. I started reading, and I love it.
Yep.
Today's post, Thursday, April 7th, last week. Malaria, Part 4, The Landscape. Great writing on infectious diseases in the environment. You would really like this, Dickson.
You're right.
Plus, on his sidebar, it says other infectious disease sites, and there we have This Week in Virology, This Week in Parasitism, This Week in Microbiology, and Alan Dove.
A standalone.
So with links like that, how can you not like this website?
You can't. You have to adopt it as one of our own.
So I highly recommend it. I've been looking at it for a few weeks now.
Fantastic.
It's really cool. And Dickson, you'll love it. And I think our listeners will like it as well. And there you have it for another TWiV. Do follow us on Facebook if you visit that website, facebook.com/thisweekinvirology. You can also find us on iTunes at the Zune Marketplace. And you can listen on your phone, an iPhone or Android device. There is an app for either one at microbeworld.org/app. Of course, you can always go to twiv.tv and play and listen and read our extensive show notes. And by the way, all our emails are archived there, so if you want to read while you're listening and visit the links that are sent in by our listeners, check that out, twiv.tv/letters. As always, send us your questions and comments. We love them. twiv@twiv.tv. And indeed, we will read most of them.
Eventually
we'll get to them. Today, Rich Condit was in studio, and Rich, of course, is at the University of Florida, Gainesville, where I am sure the weather is nicer than it is here. But good to see you here, Rich.
I'm not looking at the weather in Florida, and I can't wait to get out of here because this little helix thing that you've got is driving me crazy.
I've suffered under the stings and arrows of that one for quite a while, Rich. Now you know why it's so difficult to do that here.
That thing is really something.
It's nice.
It's great to be here.
Yeah, good to have you in studio. Before you leave, take some pictures with your iPhone, and we'll put them on the Facebook page.
Nice.
Excellent.
Nice.
Thank you.
Nice.
Dickson Despommier, thank you for coming by.
My pleasure, as usual.
I'm glad that you came to lowly TWiV after the American Museum of Natural History.
No, they're at the same level. They are at the same level. Someone asked me, why are you leaving? And I said, I have to go make a podcast now.
And they probably
you're so
said,
busy. You're so busy.
They probably said, what's a podcast?
No. They all knew. They knew.
Good to see you again, Dickson. And Alan Dove is the only one not with us today. Someday, if you're ever in New York, Alan, you should come by and do a podcast.
We'll try to get down there at some point.
You do have family here, right? Actually, north of the city.
Yeah, my in-laws are in Westchester County, so maybe
close enough.
If you ever come for a weekend, come on the Friday. And Dickson is always here now.
Always.
I'll fly up.
That'll be good.
We'll have a TWiV party.
We'll fly down.
Oh, okay.
You just stay right where you are, Rich.
Depending on the season.
And there you go. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.